Okay, well, I think we can get started. I was just waiting for last-minute stragglers, just allowing a little extra time since we always underestimate how long it's going to take us to get somewhere. Well, good afternoon, and I want to welcome you to the 30th anniversary celebration of the Tyler Prize for Environmental Achievement. Tyler Prize is an international prize which is given each year for seminal contributions in the areas of environmental science, environmental policy, energy, and human health. And it's given for contributions that basically have a major impact on humankind. My name is Linda Duguay, and I'm the executive director of the Tyler Prize. And it's my great honor and privilege to stand here and be able to introduce this year's truly outstanding and truly international 2003 Tyler Laureates, who are being honored and recognized for their major contributions within the broad area of environmental health. The Tyler Executive Committee, actually the entire committee is actually here today to participate, and I just want to sort of single them out. The large group of gentlemen in the front and in the second row, and we have Sherry Rowland from UC Irvine. If you want to stand up and just be recognized. This is, I think, and I want Sherry Rowland and Neil Sullivan, who's the vice provost here at USC for research. And then we actually have my predecessor. I've only been at this job for about 3 years, so I'll identify Jerry Walker, who actually held this job for about the first 25 years, I think. Jerry's done an outstanding job. Dr. Arturo Gomez Pompa, who is also one of our Tyler Laureates, and Arturo's from UC Riverside. We have Dr. Jim Maloney, former head of surgery at UCLA. We have Dr. Robert Frosch from the Kennedy School at Harvard. Coming up, we have Dr. Joel Cohen, another Tyler Prize laureate from the Rockefeller University. We have Bob Sullivan, who's our executive chairman. Bob, would you stay? Bob's private business, private industry back in Arlington, Virginia. And we
dialogue led us to understanding the bottled water phenomenon. And so we're seeking papers to understand how that happened and why it goes on. And we're going to give the winner a prize to be announced by Neil Sullivan with all the papers.
Yeah, the Sullivan effect. The double Sullivan effect now. We do have some very interesting discussions, as you can imagine. And we also have Dr. Owen Lin from Baylor University. Owen's one of the founding members of the committee. He's been on the committee for its entire 30-year history. Is that right? Along with Dr. Ralph Mitchell from Harvard University, who's also one of the 30-year members of the committee. We also have Dr. Judy McDowell hiding in the back row from Woods Hole Oceanographic Institution. And I always seem to forget something. Sherry's not only a former, Dr. Sherry Rowland's not only a former Tyler Prize awardee, but he's also, of course, a award winner of the Nobel Prize. So that's, didn't mean to forget that, Sherry. But, this group has a really tough job, as you can imagine, to sort through the large number of excellent nominations that we get each year. And I think they truly do an excellent job in picking extremely worthy candidates. And I think this is particularly evidenced by the 3 international candidates that we have this year and the 51 former laureates who have preceded them. I think you all, if you didn't pick up a copy, we have these commemorative booklets in the front if you'd like one. It sort of gives you a little history of the Tyler Prize, and then it also tells you a little bit about each of the laureates and for what areas they were awarded. You can see it's a very diverse group of Tyler Prize winners. But on to today's celebration. We have with us today Sir Richard Dole, Sir Richard is the Regius Professor Emeritus from Oxford, and he's being awarded the Tyler Prize to honor him for his pioneering contributions on the association between a range of environmental agents and cancer. He's recognized worldwide for his many contributions to the field of epidemiology. And I think I was sitting at his table at lunch with Malcolm Pike, and Malcolm said, he is the epidemiologist of the world. So, Sir Richard, we're honored to have you. And I think his most outstanding contribution was his seminal work establishing beyond any question the link between lung cancer and cigarette smoking. And of course, Sir Richard was a smoker and since found better to mend his ways, having made those connections. And you can see that was a good thing because he's lived to be a long and healthy life. And I think Sir Richard, through his meticulous research, has done more than anyone else to identify preventable cancers and to ensure that actions are taken to protect individuals from harmful exposure. We also have with us in the front row Dr. Hans Herren from the International Center of Insect Physiology and Ecology. Hans is the director there. I guess it's affectionately known as ECP in Nairobi, Kenya. And he's receiving the prize for leading one of the largest and most successful biological control programs in the world. His research and his action saved one of Africa's most important food crops, the cassava. When the mealybug was attacking the cassava plant and threatening over 200 million people in Africa with famine, Dr. Herren and his research team identified and introduced a parasitic wasp, a natural enemy of the mealybug. The wasp brought the mealybug under control, and it prevented widespread famine and death of an estimated 20 million people. This biological control program also, as you can imagine, eliminated the need for any risks associated with chemical pesticides. Our third laureate, Dr. Yoel Margalith, who's a professor of entomology and director of the Center for Biological Control at Ben-Gurion University in Israel, is unfortunately unable to join us today. Yoel unfortunately suffered a heart attack just about 10 days ago. It was fortunately a mild one. He's already undergone bypass surgery last Monday. He's doing quite well. My assistant, Sue Anderson's had a couple of delightful conversations. He sounds very good, very well on the mend. And of course, he's very sorry that he couldn't be here to join us. But Dr. Margalith is being honored for his contributions to also in the area of biological control, but in this case of mosquitoes and black flies. His discovery in 1976 of a new microbial subspecies known as Bacillus thuringiensis israeliensis, also affectionately known as BTI worldwide, I think, has had an enormous effect on human health and on environmental quality. BTI, again, is a naturally occurring microbial agent which is lethal to pretty much most species of mosquitoes and black flies, and unlike chemical pesticides, it has very little impact on the environment. So it's had a tremendous impact, and there's no resistance to date has developed against BTI, and again, it's been responsible for saving millions of people from the suffering of a variety of diseases throughout Africa and around the world at the moment. So without further ado, I'll introduce Sir Richard, who is going to speak to us first on getting risks in proportion, and then Sir Richard will take some questions, and then he'll be followed by Dr. Herren, who will give us his lecture on saving lives, saving the environments, the African-wide cassava mealybug biological control program. So, Sir Richard.
Madam Chairman, members of the Tyler Committee, I haven't turned me. I'm so accustomed to speaking so loudly that I don't usually use these things, but I will. Now if I talk too loudly, you'll have to say so. Members of the Tyler Committee and co-environmentalists, when some 70 years ago I started on a medical career, the hazards with which people were predominantly concerned were those of infection. They were hazards that people could do very little to avoid. Not long before, a pandemic of influenza had killed some 20 million people throughout the world, and in 1 year had killed more people in the UK than 5 years, and many in their prime of life, than 5 years of intensive fighting in the First World War. Epidemics of diphtheria, measles and whooping cough carried off hundreds of thousands of children every few years. Lobar pneumonia had a 20% fatality rate. Pulmonary tuberculosis was expected to carry off one student out of every medical student out of every intake of 50 each year. No, I don't mean pulmonary tuberculosis killed one a year, but for each intake of 50 in a year, one was expected to get tuberculosis. Streptococcal and staphylococcal infections made every open fracture a threat to life. And I might well have died myself from streptococcal septicemia as a result of pricking myself during a post-mortem had not Prontosil sulfonamide been introduced just very shortly before. Well, those days in developed countries like ours are long past, thanks to sulfonamides, antibiotics and immunization. But I mention them because they inculcated the idea that hazards to life was something that nature or other people had caused. With the conquest of infection, living organisms as the bearers of disease in developed countries like ours were replaced in the public mind by chemical and physical pollutions, epitomized by the smogs in Donora Valley and in London in the early 50s. The progressive discovery of more and more carcinogenic agents in industrial processes, from coal tar through asbestos to vinyl chloride, the increased mortality from cancer in the survivors of the Hiroshima and Nagasaki bombs, and the destruction of bird life from the widespread use of herbicides and pesticides that was described so vividly in Carson's book Silent Spring. So far indeed had the pendulum swung in the direction of blaming chemical pollution by the chemical industry for disease, that in 1978 advisors to the US Occupational Safety and Health Administration estimated that 23 to 38% of all fatal cancers were attributable to occupational exposure to 7 industrial chemicals alone. Now, this extraordinary claim, which was accepted, I may say, by the Secretary of State for Health, Education and Welfare, and was widely believed, led the Office of Technology Assessment to seek independent quantitative estimates of the avoidable causes of cancer. And they asked my colleague Richard Peto and me to produce them. This was a formidable task, and we undertook it with some reluctance. Eventually, however, we reached conclusions that are summarized, if I can operate this correctly, are summarized in the first slide.
Hold it down for about a minute. Right. Hold it down hard.
I'm holding it down as hard as I can.
It has to be turned on by hand.
Is it not on?
There we go. Yeah. Thank you. Eventually we produced these estimates. Now, some of the estimates we thought were fairly firm, most notably those for the effects of smoking, of tobacco, alcohol, and ionizing radiation. But others, particularly that for DART, were little more than informed guesses. The emphasis, however, had clearly shifted from chemical pollution to factors over which the individual had some personal control. Now, this was no new idea, for broadly similar estimates had been made by some other epidemiologists. most notably by John Higginson, Director of the International Agency for Cancer Research on Cancer in Lyon, and by Ernst Winder, Director of the Cancer Research Foundation in New York. And our estimates were not seriously challenged. In the 22 years that have passed since the publication of our report, much other evidence has been obtained, and interest, as you'll be only too aware, has come to focus on the effects of variation in the human genome, knowledge of which may, in the course of time, improve out of all recognition our understanding of the pathogenesis of disease, and hence we may hope of both the theory and practice of medicine. In the immediate future, however, it remains true that the most effective methods for prevention of cancer in developed countries like ours are those over which the individual has some personal control. Now, cigarette smoking, as in 1981, continues to be the foremost hazard, not only of cancer, but also of all causes of death, though it is one that is now coming under control. When in 1950 Bradford Hill and I found that there was such a strong relationship between smoking and lung cancer that we had to conclude that cigarette smoking was an important cause of the disease, we had no idea that it might cause many other conditions, apart perhaps from some other cancers of the head and neck. But when we began to follow up a group of some 34,000 doctors with known smoking habits as a check on our conclusion, we began to find that smoking was associated with many other potentially lethal conditions, now known to be about 40, and that for most of them the association could be shown to be causal. Now we know, both from our own studies and those of the American Cancer Association, that regular cigarette smoking causes adults to have double the mortality of lifelong non-smokers. In other words, regular cigarette smoking from adolescence throughout adult life is as dangerous as all other hazards combined. Now, its relative importance is brought out clearly by the contrast that my colleague Richard Peto has made with violent causes of death in the United States, which should be shown in that slide. Fortunately, however, the worst effects can be avoided to a surprisingly large extent by stopping smoking, as is illustrated by the comparative survival of lifelong non-smokers, ex-smokers, and continuing smokers among British doctors. Even those who stopped round about the age of 60 received some benefit. The top line shows the survival from the age of 35 onwards of lifelong non-smokers. The bottom line is of cigarette smokers, and you can see there is from just down at the bottom, the people who stopped round about 65 a higher proportion of them do get to the 80s than the continuing cigarette smoker. But of course, if you stop round about the age of 35, in our study, the survival was indistinguishable from that of the lifelong non-smoker. But in fact, some damage is done by the age of 30, and more detailed studies have shown that in the general population, who probably began smoking seriously at an earlier age than the British doctors did, there is some irreversible change, even by the age of 30. This we were able to show in the course of our study of the effects of radon in southwest England, when we obtained detailed smoking histories for 1,000 people with lung cancer and 3,000 representative other people from the same areas. These results are illustrated in this slide, which shows the cumulative mortality from lung cancer, the bottom line being in lifelong non-smokers, the top line giving about 17% mortality, which Malcolm will remember that figure well because we had to calculate it at one time in response to some criticism that was made of our studies of the continuing smokers, and the intermediate lines show the cumulative mortality from lung cancer of stopping at different ages. The one just above the lifelong non-smokers being that for people who stopped around 30. So some damage was done, even irreversible damage had been done by that age. But the enormous benefit of stopping smoking is, however, better illustrated by comparison of the trends in the prevalence of smoking and in the mortality from lung cancer in the national population. And this shows figures for UK, the prevalence of smoking, the top line of men and the bottom line for women, and you see it did begin going down shortly after the war in men, but it didn't go down rapidly until the 1970s when the media came on our side and stopped putting out tobacco arguments to counter the evidence. And then there was a more rapid reduction in the prevalence of smoking, and you'll see the corresponding reduction in the mortality from lung cancer, even now beginning to show in women, which is really very dramatic. Of course, in the United States, where cigarette smoking, the switch to cigarette smoking occurred a little later than in the UK, the increase occurred a little later, but the reduction is occurring now just as well, and it's occurring particularly well in California. There we see figures for men and for women, the lower the two pairs of lines being the reduction in mortality, the change in mortality from lung cancer in California compared with in the United States as a whole. And I needn't remind you how much more effective the education about the effects of smoking has been in California than in a number of other states where they used the money that they got from the tobacco industry for other purposes other than the purpose for which it had been intended. Now, but enough of smoking, if not too much. Estimates of the quantitative contribution of 11 other categories of cause are shown in this slide. Now these figures are recent ones which we made for the UK. They wouldn't be so very different from the United States. They differ from those that I showed you earlier that were made in 1981 in several notable ways, particularly the inclusion of obesity and physical inactivity as two separate factors, and for the attribution of a larger proportion to infection. Time precludes their detailed examination, and I will conclude by referring only to three causes, the effect of which my colleagues and I have helped to quantify. One is exposure to asbestos fibres in the air, which has constituted an occupational hazard and has contributed to atmospheric pollution, over which people have little or no personal control. Exposure to such fibres has been associated with the cause of lung cancer for quite a long time, particularly from autopsy observations of its association with asbestosis. And that had been observed for some 15 years before I was able to study a cohort of workers that were exposed to heavily exposed to asbestos in their work. And I was then able to show that they had a mortality 13 times that of the country as a whole from lung cancer. This, of course, in the early 50s, was at a time when lung cancer was still quite a rare disease. It wouldn't be possible to have had 13 times the mortality that we have had from lung cancer in recent years, as many as 8 or 9% of all deaths have been due to lung cancer, so you couldn't increase that 13 times. Later, when the dust levels in asbestos textile factories had been greatly reduced, we were able, with the collaboration of industry, to suggest a linear relationship between the concentration of fibres in the air and the risk of lung cancer. And this shows our data. Other data that were a bit closer to a straight line than our points were led to the same conclusion. But from this quantitative relationship, we were able to estimate that exposure to a concentration of 1 fibre of white asbestos crassatile per millilitre of air during a working lifetime of 30 years would produce a 2% risk of developing lung cancer by the age of 80, a figure which would of course be lower, substantially lower now, because asbestos and tobacco smoking act synergistically, so that with the reduction of smoking, so the amount of lung cancer caused by asbestos is reduced. The second cause, ionizing radiation, was something that I began to study after the first test explosion of a hydrogen bomb over the Pacific in 1954, when radioactive fallout occurred worldwide, principally in the northern hemisphere, and governments suddenly realized that they had no clear idea of what the effect of this was likely to be, other than possibly causing some risk of hereditary disease. At the request of the Medical Research Council, Court Brown and I determined the incidence of leukemia in 13,000 patients given radiotherapy for the treatment of ankylosing spondylitis, and related the incidence to the dose that they received throughout their bone marrow. And this led us to conclude, I quote, "The incidence of leukemia bears a simple proportional relationship to the dose of radiation, and that there is no threshold dose for the induction of the disease. Calculations lead to the conclusion that the dose to the whole marrow, which would have doubled the expected incidence of leukemia, may be within the range of 30 to 50 rad, or as we would say now, 300 to 500 milligray. Further observations, of course, showed that ionizing radiation could cause cancer in practically every organ, as of course was shown by the observations on the survivors of the Hiroshima and Nagasaki explosions, and as we, along with many others, also showed from studies of patients irradiated. But we were able to show that the direct effect of radon in domestic houses on the incidence of lung cancer by the study which I have described previously, and in our observations led to that relationship, which is much the same as has been found in studies in the United States and in several other studies in Europe, all of which are now being pooled together in order to get a really precise estimate of the amount of lung cancer caused by radon. But from these results, we estimate that about 6% of lung cancers in the UK are due to radon. It would be 12% in the United States, because radon levels, generally speaking, are higher in the States than they are in England. But of course these are nearly all in smokers, because radon and tobacco smoke act synergistically, just like asbestos and tobacco smoke did, so that in the absence of smoking, the effect of radon is very small indeed. And our third cause that I want to refer to is steroid contraceptives. In the mid-1980s, Vesey and I helped to establish the risk of venous thrombosis associated with their use. And I must tell you this story because it's the only pleasure I get out of it. is telling people the story. Some lawyers in America wanted me to come and give evidence about how steroid contraceptives had caused a woman to have a thrombosis in her leg and lose her leg. This was in Texas, and I didn't want to go to Texas to give evidence. I didn't really think it was the Kemp pharmaceutical firm's fault. And the lawyers said, look, you just tell us how much you want. We want you to come. Tell us how much you want. And I said, you haven't got enough money in Texas to enable me to come. We shut him up. After that study, we tried, like with many others, to see if it had any relationship on the risk of breast cancer, but the risk being very small, we and all others who studied it got contradictory results. And it wasn't really until under Valerie Burrell's leadership that a collaborative group was organised in which Malcolm Pike has played a leading role, which has brought together practically everyone who had worked on the subject. And Peter and I have also been part of the organising committee. An analysis of the data for 53,000 women with breast cancer and over 100,000 controls showed that steroid contraceptives caused a small risk of breast cancer, illustrated on this last slide, 24%, but just whilst the contraceptives were being taken, and within 10 years afterwards the risk had essentially disappeared. As they're mostly used by young women in whom at which age the ordinary risk of breast cancer is very low, the risk is quantitatively not very great. Now, if I could just have all the slides off. In conclusion, Madam Chairman, I have, as requested, talked mostly about investigations in which I've been personally involved, and have in consequence not been able to show, as I had hoped to be able to do, the relative unimportance of many factors that cause so many concern. I hope, however, that I have given some idea of the relative importance of a few factors, and above all have indicated that work in this field, that is in determining the relative risk of different activities, is not something that the individual these days can readily take on by him or herself, but that it requires collaborative effort in which many people work together for the common good. Thank you.
Sir Richard, that was truly excellent, and I want to thank you for that excellent lecture. And I want to open it up to maybe just a few questions. I think I see one back there. Dr. Manahan?
Is it true that the great statistician Fisher was brought in this debate early on about correlative studies and was turning things on its head, saying, cancer causes smoking, et cetera, et cetera? I remember hearing that as a student. Is there any truth to that?
Oh yes, he wasn't brought in, he came in voluntarily.
I wondered if he was brought in by the tobacco.
No, he wasn't. He did smoke a pipe, but he wasn't influenced by the tobacco industry, that I'm quite sure of. But he was particularly upset when an article in the British Medical Journal after a second paper said, this is something which has got to be brought to the attention of everybody as extremely important, and everybody must be told about it. Well, he detested the exaggerated propaganda during the war, and he thought this was propaganda, and this irritated him greatly, and he put forward this alternative hypothesis that some genetic factor caused both people to smoke and to develop lung cancer. Not, to my mind, a very attractive explanation, particularly as it wouldn't explain in any way the increase in the disease. You then had to say that was all an artifact, despite the fact that it increased 50 times and differently in the two sexes. Eventually, he did also point out that in our study we had found that the risk was slightly greater in people who said they didn't inhale than people who said they did inhale. And he said that really we ought to conclude that inhaling was an important protective factor. Eventually, in larger numbers, what was shown was that inhaling was positively associated with cancers in the distant part of the lung and was negatively associated with cancers in the upper bronchi, which was not altogether unreasonable. I'm told by Sir Walter Bodmer, who was a pupil of his, who visited Fisher just before he died in 1962, I think, that he had intended to write and say that he recognised that smoking was a co-factor. He didn't go so far as to say it was a cause, but it was a co-factor in the production of the disease.
Okay, I think we'll take two more questions, right? One right here.
I'd like you to, the big number that you didn't talk about on those slides had to do with diet, and I'd like to hear a few words about how you can come to the conclusion that so many cancers are due to diet, and what does that mean?
Have I got another half hour?
I think we might have to have that as a sidebar. I think Dr. Herren has to have some time.
Now, I really can't answer that briefly yet. The evidence on that is each year it gets messier. And we're really going to have to do controlled trials with individual materials to work out what are the components. But I think there can be little doubt that a diet of a developed country does play some part in the greater incidence of cancer than of the large bowel, for example, than the diet in more in undeveloped countries. There's pretty good evidence that cancer of the stomach is related to salt-preserved foods, and is disappearing quite rapidly with the disappearance of salt preservation and the reduced consumption of salt in Japan and China. But apart from, well, of course, there's a clear evidence of aflatoxin, the fungal contamination causing cancer of the liver, and there's clear evidence that overconsumption causing obesity is responsible for a very substantial proportion of cancer, estimated now to be 10% of all cancers in non-smokers in the United States.
One last question, Neil.
Yes. These days, interdisciplinary research is considered by some to be a buzzword, and others sort of the direction of which research will be going in the future. And I thought your very last sentence of your presentation began to address that. Do you have perspective that you could share with us on a long and successful program of research of your own? What do you think of these ideas?
Well, this has been talked about for some time. It hasn't as yet, I think, revealed anything very striking. Maybe when we know a bit more about the genome, we shall find about how it interacts more with some components of diet, but I'm not very optimistic about that. I have to admit that what I meant by my last sentence was that you needed large numbers, and you need people in different situations that one research group wasn't really able to deal with, like the EPIC study we have at the present moment in Europe, was groups studying diet in Sicily and in Finland and in the UK, where you really then do get big variation in diet. That's the sort of cooperation I had in mind.
Thank you, sir. Oops.
As a university professor from Texas,
I should have said that he was from Texas.
I certainly do not have the funds to encourage you to come to Texas. As a
from Houston, isn't he?
Yes.
He visits voluntarily. He was just in Houston before we arrived yesterday.
Yeah.
Thank you, Sir Richard, for that
Executive Director, members of the Executive Committee of the Tyler Prize, fellow scientists, friends, and also family. It's a real pleasure to be here today with you and to present a bit of the work which I've been carrying out with many colleagues in Africa. The little paper today, so Saving Lives, Saving the Environment, the Africa-wide Cassava Mealybug Biological Control Program. I will talk a bit about this, what we did and how we did our work in Africa. Oh, I need to go there first because... Okay, sorry. I thought I'd mention something first about the institutions where I was working in so that this is very clear. This Cassava Mealybug program was done at the International Institute of Tropical Agriculture in Ibadan, Nigeria, and later on for the same institution in the Republic of Benin, as we had some difficulties operating in Nigeria, and anyone who has been there will probably understand why we then moved to another country to be able to really do our work across the continent. And the IITA belonged to an organization called the Consultative Group for International Agricultural Research. And this is a system which is run out of the World Bank, which links 16 research institutions around the globe together in what is called the CGIAR, or also commonly called the CIGAR system. Although I did not really work on tobacco. And you may know IRI, the Rice Research Institute in Manila, or maybe CIMIT in Mexico, which are two of the sort of the most common institutions of this system. And give you an idea also what kind of money we're talking about. That whole system runs at about $350 million a year for all the research which is done in the international ag sector in the tropics. And this level has been basically stable for the last 20 years. Actually, it's dropping, not even going up, it's dropping, as you probably know. And the mandate is in the area of research, capacity building in the main staple crops and livestock. And now we heard about ICIPE, and ICIPE is the International Center of Insect Physiology and Ecology, based in Nairobi, Kenya. We are not a member of the CGIAR directly, but we have our meetings same times, also the same donors group, but we are basically an institution by itself, somehow coming under the UN umbrella. And again, same mandate, research, capacity building. We do also some development in the areas of human, animal, plant, and environmental health. And as the title says here, it's always linked to insects. And I switched from IITA to ICIPE in 1994, but the work I was talking about now relates to what I did while I was working at IITA. Now, cassava is a major staple crop for 200 million people, or even more, 250 million people these days across the tropical belt of Africa. And right now there's more than 10 million hectares or 20 million acres under cassava in the humid tropics of Africa. And you can see 72 million tons, so it's a very important crop. People in the zones, in humid tropics, eat cassava for breakfast, lunch, and dinner in many different forms. Not only the starchy tuber, but also here, this is the part which is eaten. It's a root, a starchy root, of which you may know one of the products here, tapioca. So if you have had a tapioca, it comes from cassava. It's the starch extracted from the cassava root. It has been decried as a lousy food which has cyanide in it and killing people. But it turns out that people actually know very well how to process it and to eat it. And without cassava, there will be major famine across the continent. In particular, in years where there's drought, because cassava is extremely tolerant to drought. You can see the value of this crop. Never mind that as a quality, well, it's like eating only potatoes. You wouldn't be that a healthy diet either. But cassava, really, I think, is really the main staple in the humid tropics, and it's prepared in many different ways. But usually people harvest it, and after harvest you have to process the root immediately and make sort of a flour out of it, slightly fried. And then you just mix it with water, cold or warm, and it becomes sort of like a mashed potato, sort of a mushy meal. But again, there's dozens of different recipes. The leaves are also eaten as a spinach, a very high content in protein. But again, also they're also cyanide, so they have to cook it long enough to make sure that they don't they die if you eat it. And there's been accidents before in Africa when people who were used to eat sweet cassava, which doesn't have cyanide, were going into a region because they become refugee, and then sort of saw cassava somewhere and ate cassava which actually had cyanide, and obviously that's not very good if you eat fresh cassava with cyanide in it. I mean, as you know, that's a very good killer. So just so there's a background to cassava itself, so it's a very important crop. Now, a few words on maybe one more. The cassava actually originated in South America, in the Amazon basin, and was brought to Africa by the Portuguese when they were going back and forth some 300 or 400 years ago. Now, the mealybug, you see it there. These are two females. There's no males known in these insects. In this fuzzy material in the back, that's where they lay their eggs. And they have a fecundity of some like 400 to 900 offsprings within 2 weeks. No males, so every offspring produces again so many. So you can see how such an insect can become a real problem if there's nothing there to actually keep it in check. So here we're showing you the area of potential areas where the mealybug could have come from. Because when the mealybug was first found in Africa, it was the specialists who identified it in the Paris and London museums. So they said that this insect is not from Africa. That's all what they knew. The other thing they sort of deducted was that the closest relative lives in Mexico, then in Yucatan on cotton. And so we had a new world species in front of us, and which would have come anywhere from Southern California to Argentina, where we have also wild cassava, and also where mealybugs, which were sort of related to this mealybug, occurred. But the closest one was in the Yucatan Peninsula. Sometimes in the late 70s, we don't know exactly, because the first occurrence in Africa, where the red dot is, was discovered in 1977. And that was just within 1 year, huge area just basically lost all its cassava due to that mealybug, just exploded and then killed the plants outright. And so, again, from somewhere in between there, that's where the mealybug originated. And within about 4 or 5 years, it had spread throughout the potential areas where it can survive in Africa, which is the cassava belt, which is sort of the shaded area there, as you can see. So very quickly, the mealybug spread, and it spread very fast because of a few things. First of all, the way cassava is multiplied, you don't use seeds, you use a piece of the stem. And by planting the stem, you carry all these insects which sit on the stem in the bud, and so it was carried around very widely, because people, they visit their friends, family. As you know, in Africa, people travel all the time, and not only within countries, but across borders. Certainly when you see in Zaire, they go to the Congo, down to Angola, so there's a lot of traffic there. And also a lot of the Zaire, so at the time it was called Zaire, now it's the DRC, a lot of Zaire were working in harbors in Dakar and also in Lagos. And the tendency of every time they travel, they take the cuttings with them, because they want to plant the same variety as they have back home. But also they take leaves, because the Zaire used to eat a lot of leaves, spinach. And so you can see that very quickly the mealybug was all over the place. Within a few years, it went all the way from this Zaire all the way to Dakar on one side and Maputo on the other side of Africa. And what you see, you've seen the field before. Cassava is about man's tall, thick canopies, and that's what it ended up to look like. And And why do I say there's no more planting material? Because those little sticks here are dry, and so the farmers didn't have anything to plant again the next year. So it really decimated the whole production almost overnight. And this is another field. The first one was in Zaire. This is in Malawi. So as you can see just how cassava was destroyed. And huge colonies of mealybugs in the top of the plant, and that's why the plant then sort of started to rot and dry. And these are all mealybugs, hundreds of thousands. I mean, we stopped counting actually, because just loads of them. And you look at a plant, a normal plant, which we sort of protected from the mealybug with all the roots here. And this was a plant which was attacked by mealybug. You can see there's nothing there. This is just the planting stick here, but there's no root, nothing to harvest. So you can imagine in the Congo at the time this happened, prices of cassava were just multiplied by 70 or more within a few months after the occurrence of the mealybug, because everything was just gone. Now, what can we do about it? First, we had to understand the size of the area where this mealybug was doing its thing. And I always like to look at the United States here. So the area where cassava is grown in Africa is 1.5 times the United States. And I mentioned this to the donor community when looking for money, because when you have a fruit fly problem in California, somewhere here looks like a small dot, like a pinhead, you spend $150 million overnight, right? Nothing is too small or too big to deal with it. And I was looking for $30 million for 10 years to solve the problem across the continent. And I was told I was a megalomaniac by the U.S. representative at the FAO in Rome. So I remember that all the time. But you just can see what you're facing. And here, this is the team I put together after 2 years. I got the first money. Dr. Noel and Dr. Kim Lema from Zaire were the two first scientists I was able to hire myself here. It was in 1981, after we got going slowly into this program. And here was really something, the signing of the first money, the real money we got to actually execute this program, representative of the International Fund for Agricultural Development, and thus the Director General Hartmans of the IITA at the time I was there. And back there in the corner, looking over the signing of the agreement, which really allowed the program to start. And that was about $250,000 to kick off the program. Now, what is biological control? Well, it's basically using what nature has given us. And normally, insects will sort of fluctuate in a certain environment, a certain low level, below what we call an injury level. We don't even know they are there. I mean, they are there. We see them here and there. Nothing really bad happens. But if you don't have any natural enemies, population goes above what we call this injury level, and we have this big damage. And the mealybug was clearly an example of an insect which was in a new environment. There were no natural enemies at all. Nothing local is even adapted to it. Certainly not in the first few years. And so we had these tremendous problems. And the idea was to go find out where this insect came from, classic trick, and then look for parasitoids, predators, take them over, release them, and see what happens. And that's what we set out to do. But it wasn't that easy either, because there were a lot of skeptics around. People, they said, biocontrol, well, let's do host plant resistance. And with cassava, this is very complicated, because it takes you 10 years to go to create a new variety. We didn't have 10 years to wait, right? I mean, things were just disappearing left and right. The older alternative, which was presented at the time, were pesticides. So basically, that was the thing to do. And 24 years ago, the pesticides were maybe not as safe as today. I mean, we have a whole lot more safer materials today than then. And the stuff they wanted to use was really quite awful material they wanted to use on cassava. And I can tell you then, and I don't know if Sir Richard got some threats, but certainly I got threats from the industry when I said, look, we don't want any of that stuff. We're going to do it the good way. But that wasn't really in the cards for many people who saw this as a fantastic business. Don't forget, 20 million acres you could use a pesticide on. Oh, that's quite something. Now, what we set out to do then, we went out and looking for this mealybug, because it was anywhere somewhere between Southern California and Paraguay. That's basically the clue we had. And what I did then, I searched in the botany books where has cassava originated? Where are the center of origins? And there are actually 5 main center of origin for Manihot esculenta. And there's up here, there's one in this area here, and then there's several further down in South America, and certainly also one down in the lower part here. And so what do you do when you go and look? So you take a car and drive around, look for wild cassava, and since people have been working on cassava for at least 80 years in Latin America, because it's not something new. But no scientist had ever seen that mealybug. So I think, why? It's sort of funny, right? I mean, so if it were somewhere on cassava, it would have been found. So my theory was that it's on a wild cassava. And many of the wild manual species are very nice ornamentals. And my idea was that somebody must have picked up something somewhere, an ornamental, and carried it around, like people do all the time, right? Oh, this is a nice flower, a nice bush, let's take it home and plant it in the backyard. So that's why we searched in the areas where of greatest wild diversity. As it turned out, it was found in spot number 5. And in sort of being from the north, what do? I had marked them 1, 2, 3, 4, 5, and started number 1. Well, we go from north to south, right? You don't go from south to north, or somehow. Because if I had it turned around the whole thing, probably would have found that mealybug within a month, not within a year of the exploration process. But anyway, that's sort of the story behind it. But eventually it got found in the border area between Paraguay, Bolivia, and Brazil by a colleague of mine with whom I was working, because I had to contract out some of the activities here. Couldn't be all over the place, and we had an emergency over in Africa. And so a colleague of mine working at the Centro International de Agricultura Tropical in Cali, Colombia, went to Paraguay, in spot number 5, visiting his former Peace Corps friends. When he walked around, he went to a research station and he knew what we were looking for. And he stumbled over a cassava field where there were mealybugs. And obviously there were so few there, that's also where then the natural enemies were found. So you can see, so basically you go out there and just look at every plant, see if there's any mealybugs on it. And so that's how actually the mealybug was found in spot number 5, which are described basically just from literature. So this, and so what do we do next? So we can't just take mealybugs or natural enemies across. So from the area where they were found, we set up teams to search for natural enemies. And they went to a quarantine laboratory in the UK. We could not take anything straight across because we didn't want to bring any diseases, for example, or any plant material. So we established in London, the Imperial College, a special greenhouse where we took cassava material from Africa, brought it there, raised the cassava, took mealybug from here, raised the mealybugs there, took the ladybugs and the parasitic wasp, and reared them for up to 20 generations before taking them out to Africa. Make sure they had no diseases, and etc. So to make sure, and then we took them down to Nigeria, where they were then multiplied and released across the cassava belt. Now this is the champion here. This is one of the more than 20 different natural enemies which we found. And Epidenocarsis, or they keep changing their name, Aponagyrus lopezi, which basically has now single-handedly controlled the mealybug for some 20 years across the continent. Maybe you should have given the prize to Lopezi, but I'll do something for it. And here we have a couple of ladybugs, which we also found when we took across and released. But it turned out that they were not very effective. And what we did also in the quarantine laboratory, we tested them to make sure they are very specific, because we didn't want to bring something in who all of a sudden goes and attacks some beautiful butterflies or something else. And as you know, the rules of the game in biocontrol actually were quite tough, even then. And now they become even much, much difficult. And I think today it will be a whole lot more difficult to do such a biocontrol program because of the new regulation, which has been imposed on the people who do biocontrol by the biotechnology lobby, basically. Because they say, if we have to test our GMO, you guys have to test your things better also. Sort of, so that dialogue is now going on. So we had found these natural enemies. We knew they were working. We knew they were attacking the mealybugs. So what's next? You remember that map there, how big the area was. And we could not do biocontrol as it was done in the past, small cages, little fiddling around and sort of, no, we had an emergency at hand. People were dying from hunger in many places. So we needed to do things big time. And again, here the megalomaniac came in. So I decided to build a big factory, a real factory to produce insects. And eventually it turned out that it wasn't something like 7-story high, but these are very modern Teflon greenhouses, fully air conditioned. And this is what we call a cassava tree. So these are cassava plants in a sort of a stem here, which has hydroponics. And so we grew the plant. And then I put mealybug on it. We multiply mealybugs, large quantities. And when the trees were loaded with mealybugs, we then put the sleeve over and released a few wasps in there. And they started to multiply those mealybugs. And then we were collecting the wasps once they were emerging from the first generation. We collected them in those little tops in containers. They were packaged in many different ways for ground and aerial releases. And we produced millions of these wasps like that to release them throughout the continent. And here the packaging, this was done either in this form here for ground releases. And here you see one. This is one of the very first releases we did in the Republic of Togo with the head of the Plant Protection Services. Because what I didn't mention, which was a fairly big problem, is to get permission. The mealybug got in without permission, right? Somebody had in a suitcase, they brought a few cuttings in somewhere and planted them. But when it came down to bring the natural enemies, first it took me almost 6 months to get the permission from Brazil to export the beneficial insects out of Brazil. Even 20 years ago, they were sitting on the biodiversity. Hey, this is our biodiversity, you know. And all right, so but I said, "Bah, what about the mealybug? You didn't really tell anyone to keep it there, right?" So now let this one go. But I spent weeks in Brasilia negotiating that we could go around Brazil and explore. It was so you can see the amount of problems we were facing. But then, once we brought them into Africa, first we had to get a permit from the OAU, the Organization of African Unity, Phytosanitary Council. And they had to be convinced that these insects will not do anything bad. I said, well, you know, really, I mean, I can say they're specific. We tested them in the lab. They said, but now we want to know that they're going to be effective. I said, oh, this is not possible. We'll have to release them and see. And after several meetings, discussions, months gone, eventually we got the permit to bring them into Nigeria, into the labs, and also for release. But in every country, we had to get a new permit for release. And I can tell you, even now, 20 years later, some countries have never given a permit. Not worry, they got the wasp anyway, because they fly over the border easy, so without problems. And then we decided that, I mean, if you fly over Africa in a big jet is one thing, but if you fly over Africa in a small airplane, you realize how big the place is. And I said, how are you going to go this? On donkey back? No. On motorcycle? Or even on car? No way we can reach all these fields. We need to do this in a modern way. So not only have we developed a mass breeding operation which was really unique in terms of biocontrol, and this was in Africa, not in Europe or America, but also we developed an piece of equipment in an aircraft. So we were flying top cassava fields. This is just for the picture, but usually flying a bit higher, and shooting out these insects, which are only about 2 or 3 millimeters long, at about 400 miles an hour. And we developed a machine, which is in this aircraft. Sorry, I don't have a good slide of this thing, which was developed in Austria, and the basic pilots could shoot out the insects. And the precision of this releasing was so good that flying over our campus, which has a swimming pool, you could actually drop a whole load of insects into the swimming pool. So you can see the precision of the release. And so we marked Africa out in sort of squares, and started to release systematically these wasps across the continent. And that was really, as I said, the battle I had within the institution And with the donors, to actually have an airplane to do this was tremendous. Why? We in Africa, why do an airplane? You know, so like this was something, I mean, hey, crop dusters, right? All over the place, but in Africa, why not? So you should see the troubles we had to actually get things done. Eventually, well, we did it all. And so here you have sort of the cassava belt. This is the area where basically we had infestation. Basically, so the whole cassava belt was covered with mealybugs. The thing spread so fast everywhere that there were hardly small areas where maybe we didn't have any mealybugs. And so these are the releases. So what happened is that we were quite surprised. After the first releases, this is the area where we had the research institution in Nigeria, we released them, and we thought that was going to take years to spread from a release point. So actually the first year, after the release, I went out with some students to check how far did these things go. So we went on foot. We thought, half a kilometer or something. After half a day walking and still finding the wasp, we said, boy, let's go back now, take the car. 150 kilometers out from one area we released, you could see a circle of 150 kilometers around where you could find that wasp. Unseen in the annals of anything. I mean, certainly nobody has done this or had such results available. And so then we had the idea, this is great, because if that's so, we don't have to release very, very close together. We just can take sort of a pattern, where is the cassava grown, and we're just going to release sort of from the air, and that's what we did. And every release point, every place we released, even 20 live females, you had establishment, which is actually fantastic. And eventually, everything was covered. And today you can go anywhere where there's cassava, you'll find mealybugs, and you'll find the wasp in equilibrium. A few exceptions here and there, and I found on the Kenya coast, for example, you do find some places where the weather conditions are so that cassava doesn't grow very well, so you would see a few little plants which show some symptoms of damage. But overall, I think to more than 90%, the problem has been resolved with this wasp. And what happened really is the year of release, so we were looking at the population levels, so you can see very high, but it was high before. Year of release, population down and stays down, just systematically down. And these are a few little peaks here, again, due to some areas where if there's a drought, you'll see a little insect coming back again and then dropping off. And this is due to the physiology of the plant, which influences the mealybug, which influences the wasp. And we have figured out exactly what happens there, also why this is so. And we know that everything goes back into balance very quickly. And here, what the result is when you're finished. Now, another part of the program is not only that we control the mealybug. That was something we did. I think it's a good thing. But training and capacity building. We have trained in this program 1,200 people. From the technicians to PhDs. And myself, I had 6 PhDs trained under this program. Unfortunately, 2 died of AIDS and 1 in civil strife. So only half are left. And you see that this shows just some other problem we have in Africa right now. But so we spend a lot of time training people and showing people, okay, what is biocontrol, how does it work, etc. And that was one of the, I think, major operations we did. In addition, and here as a group of young scientists which go to the field, been trained in doing sort of post-release sampling. And this was done in 28 countries. We had teams set up to do this. And we funded them all out of the headquarters. So funding was going to this national program that had motorcycles, teams, following up what's going on. And these people now are there. They are doing now biocontrol of mango mealybugs. They are doing biocontrol of maize stem borers and God knows what. So you can see this something established. And somebody was very happy by the donors. Cost, benefit. Now you show this to the guys in the World Bank, they will say, oh, this is great. Because $1 in, 243 out. Now you remember the number about the CGIAR. It was calculated that this program has paid the whole cost of the CGIAR for 20 years over. So basically the return just of that particular program here has paid basically for 20 years the whole system. Now, why is there no money to do research these days? You can ask yourself, and this is really something I'm asking, but So that's really something we managed. And I think at the end of the day, which is even more important than the World Bank guys and USAID and others, is that the farmers can have a crop, the families have food, and I think we allowed basically a crop, which is a key crop in the African environment, to survive. And maybe just as a conclusion here, so some of the main outputs. So I think biocontrol has not only solved the cassava mealybug problem, but has led to many new programs. Another one which I started at almost the same time is the cassava green mite. That one has been finished just a few years ago. Same success. It wasn't just a big problem with the mealybug, but quite a problem throughout the cassava belt. And again, the cassava green mite also came from Brazil, and that's where we found the predator. Mango mealybug came from Asia, been controlled also with biocontrol. Through those teams we have set up. So they just keep going and doing more things. Maize stem borers, same thing, biocontrol. Stem borer from India to Africa, also controlled, again with the same technology. You can see, so once you train people, they can keep on going, doing it. And now we are trying to do new things, in particular also in disease vectors, for example, tsetse flies and malaria and mosquitoes. The training, mentioned before, is very important, so that there is now a crew trained. And many of the people we train actually end up now in higher position in government. So it helps also on the policy side, which is very important, because if the policy environment is not there, nothing will happen. And I think that also it shows that there are alternatives to classical pest management, which means old, chemical, or even the new ones like biotechnology. Because right now there's a lot of push to use all these GMOs, but we really feel very much at our center that it's not going to be a silver bullet as it is proposed. It's going to create the same problem as the chemicals, because it's a single solution. It's not an integrated solution. And I think this is very important. Well, in the framework of this Tyler Prize here, is that we really avoided the utilization of a huge amount of pesticide. And at that time, the pesticide they were using was a systemic pesticides, which was applied on the ground, taken up by the root, and a systemic insecticide. Because mealybugs, they are protected by this waxy coat. And if you know how people harvest cassava, there's one plant today, another one tomorrow. So you don't have these waiting periods. If you use this in your field, well, how are you going to wait a month until the pesticide has gone away? You need to eat every day. And so there were a lot of problems, and I think we managed to avert a major disaster in terms of also health and the environment, because of the pesticides at that time, extremely toxic. And again, I think the importance is to maintain biodiversity and environmental integrity, because when we went back to the Americas, well, we are lucky that cassava was still around. If CD had taken over there and things would have been gone, who knows? We would maybe not found a wasp or a predator. And the millions of lives, I think, which we were able to save across the continent, I think was also very important. And also the crop itself, which is maybe not the best crop in terms of nutrition, but it's certainly something which people can count on to be there when they need it. Thank you very much.
Well, thank you, Hans. That's certainly an incredible story, and I think most probably have a couple more questions. But before we take the questions, I just want to say we're going to have a reception that's going to be 2 floors down in the vineyard room. If you go down on the right-hand side as you go down, so you can meet Sir Richard and Hans later and talk with them about these exciting areas that they've been working in. But questions? Yes, David.
So you mentioned the need to preserve biodiversity and environmental integrity, but as we know, a lot of biocontrol programs have done exactly the opposite of that. So what kind of precautions do you need to have to avoid those kinds of problems, especially in tropical islands and habitats that really have no way to rebound from introduced species?
Yeah, that's a good question. I'm yet to be shown one example of insect biocontrol which went wrong. I mean, people have been trying to do all kinds of things with biocontrol, snakes and other vertebrates. But with insects, I think we have taken a lot of precaution, and we're being very, very careful. Also, insects, they are much more specific. I mean, sure, you have sort of very polyphagous insects also, but I think if you really restrict yourself to do things in a very good way, scientific way, I think you can avoid problems. And I think that the fact that I mentioned earlier that we need to tighten, or the guidelines, the regulation has been tightened up in the last few years on biocontrol, may not be a bad thing, but it's just a whole lot more costly now to do biocontrol programs than it used to be in the past. But I think the whole solution relies into doing very good research, looking at what's happening really in the area of origin. And the chance we had in this program, which was unique, at the time we had a lot of money. I mean, it was hard to get started, but once we started, the first success came. We actually had enough funding to do very good research in the area of origin. We are doing good research in the quarantine labs and also in the labs in Africa before releasing these insects. And so that, sure, you always take a risk. I mean, I think that's why I mentioned you give me a permit, I'll tell you, all right, there's 1 in a million that something goes wrong, or 1 in whatever, but I cannot give you just a 100% guarantee. Now, what do? You use the pesticides, you introduce a predator, natural enemies, or I think it's also to make a proper choice and looking at the pros and cons of different methods. And so that's what one has to balance out all the time, whatever you do, because there's nothing. I said the only way, sure, not to make any mistakes is to do nothing, and that wasn't an option.
Front?
Has anybody thought about whether there's some way to make a business out of this?
Yeah, they are.
As a way of increasing, among other things, the rate of its use?
There are actually quite a few businesses in biological control, people producing predaceous mites for use in apartments, indoors. There are in greenhouses in Europe, there's many companies here also in the US who produce beneficial insects for use against major pests. The example of what we did, you can't make any money with this because you do it once and then it goes on forever by itself. And what people do is they raise some of the insects like trichogramma, like egg parasitoids, which you have to put out every year because they can't survive for all kinds of reasons. And but with this method here, that's the big problem, and that's why nobody likes this very much, because, well, you do it once and then you're out, you do something else.
I wasn't thinking of going into the wasp business. I was thinking of going into the insect predator business.
Mm-hmm.
So that you don't do the same product all the time, you just keep putting out new products. There's a large parade of predatory insects and diseases.
Yeah, I think we should do a whole lot more, and you're right. I think there are many new options which are possible. Actually, there's some efforts in the US. I think it's in Phoenix. They're trying to breed new predators. I mean, sort of genetically modified, which are more aggressive, who can do different things, or ladybugs, which cannot fly, so they don't get out of your field. So if you put them on your roses, they'll stay there. They can't fly away, because it's all the problem, right? They will put them there, and tomorrow, where are they? They're gone. Find something else. So there's all kind of things people are trying to work on now to see how can we make a better business out of it. And I think that a bit of more science and support in these areas, I think, could come up with some very good idea on how we can even make money and doing the right thing also altogether. Yeah.
Neil, have a question?
I had 2 questions, actually. One was, what is the genetic diversity of the cassava crop in the belt?
There are, if you go back in this area of origins, there must be something like more than 80 different wild varieties. Now, in the crop itself, it's very wide also because, I don't know exactly why, but you have the sweet cassava, which have actually no cyanide at all in the root, and the very bitter varieties, which have a lot of cyanide. So there's a lot of variety of diversity available. But it seems that from all what we have seen, they are almost all equally attacked. There was a slight difference in the very hairy cassava. So some cassavas, they have the young leaves are very fuzzy, a lot of little hairs, which the mealybug doesn't like very much. So the breeders at the very beginning thought, well, no problem, we can just select them out and do something. But as it turned out, that when the leaf grows, obviously the hair become less dense, and the mealybug goes at it anyway. And because you need only very few mealybugs on a plant to actually to start, it starts to curl, because the mealybug injects a toxin into the plant, which makes it very curly, and protects that mealybug inside there from the predators. It just covers them up nicely, and then they start to breed in there like crazy. So I think so very few millibugs actually will produce also a damage. So that's why on that part of sort of using diversity and resistance, well, it wasn't really panning out very well.
The other question was more of a philosophical one. When you run a program like this that goes so far beyond science to involve operations and logistics and fundraising and policy, and has had a profound effect on feeding people in a number of nations where population could be argued to be the basic problem. How do you think about population control, just philosophically, when you are having such a positive, profound effect on keeping people who are otherwise starving alive? Do you get into that in any context?
Yeah, I mean, I've been asked many times. First of all, why do you work such a lousy crop? Why do you do this? That crop should have disappeared. Should let it die because it's no good. Let's plant more maize. I mean, I've been that many times, and I really fought that side of it also. But then on the population side, as you know, what breeds people is poverty, and actually, so I don't think by having less nutrition, you have almost less people. I think it's the other way around. And I think there's some studies have been shown that there are more people there before there's more food, not the other way around. It's a very interesting paper which came out about 2 or 3 years ago, which I want everybody to, I get all my students and so to read this, because I think we started in the wrong way here. And because people are poor, there's more people, because this is in Africa and in many other places. It is a life insurance. You have 10, maybe 2 or 3 make it. If you have only 2 or 3, well, they're all gone. And I mean, I've seen this so many times in families of our workers who somehow have only 2 or 3 kids, but they go. I mean, you know that for every 30 seconds I've been talking to you here, a child has died of malaria in Africa. Every 30 seconds. Doesn't have to be. And that's why I think people somehow have more children.
One last question, Arturo?
Yes. Can you comment on this trend that is happening in many countries of the diminishing the number of students and schools of agriculture, the interest of new people in agriculture? Is that also occurring in Africa? Or what do you think about it? It sounds like a very strange process, but it's happening.
It's happening, and I think it's going to be coming back, visiting us very quickly. I think because we have too much food here in America, in Europe, we don't know what to do with it. We dump it. We don't know what we do with it. Make alcohol, God knows what. And so basically, the people who give us money. I mean, the citizen in the street here. You go ask for more money for agri research. They say, why? We got plenty of food. And they think that the technology can just be transferred down south. It can't. And what's really a big problem, and I've been looking into this, why could we not have more agricultural schools? Well, like, in Honduras, they have the Zamorano school, which is a very good example of a very practical agricultural school where you train farmers, and you train trainers. But in Africa, there's very few, almost none. And every technical college, which started some 20 years ago, today is become a university. Nothing against universities. But I think it's on the wrong path. They can't even maintain them. But the technical schools, where people can learn the skills to use modern agricultural tools. Now you can see the Monsanto and others. They come to Africa and say, now here guys, get GMOs. That will solve all your problems. Nonsense. First of all, the farmers can't read. Their soils are depleted. So that seed is not going to do anything. So all of a sudden, all the research, the little research we've done in Africa, should be done on this biotech stuff. Wrong timing. And I think that we need to train the farmers to become more literate about farming, about maintaining the soil fertility, about using modern tools. And I think we should bring in more information technology. I mean, today we can predict next year is going to be a dry year, right? We can predict this, more or less, or rather wet. El Nino thing. This is no longer really very difficult to do. And the farmers will be very much helped by having this information, so they can choose to plant sorghum, cassava, instead of maize, if it's going to be a dry year. If it's going to be a wet year, well, plant more maize, because it's more money, it's maybe better food. But this information is just not going to where it should go. In the same time, a lot of the resources are used to make BT maize and BT cotton and stuff like this, which is not going to do a thing for many, many years to come, because they don't increase the food quantity. They make maybe production cost less, but that's good here in Europe. In Africa, who cares?
Unfortunately, I think it's getting late, but why don't you join us for the reception, and you can maybe ask Hans and Sir Richard the questions that